Welcome back to another episode of Syntalks, a podcast which explores the field of synthetic biology. This student-run podcast is organized by the Adjunct Toronto team, an undergraduate research team hosted at the University of Toronto that focuses on scientific research in the field of synthetic biology. I am Kai, the host of today's episode, and I'm joined today by Professor Keith Pardee, an assistant professor in the Leslie Dan Faculty of Pharmacy. He is also part of the Canada Research Chair in Synthetic Biology and Human Health, as well as Medicine by Design Investigator. So welcome to the show, Keith.
Thank you very much.
And would you mind just briefly introducing yourself and giving an insight on what your current research interests are?
Sure. So our lab is at the Leslie Dan Pharmacy building and our faculty. And we work in the field of synthetic biology, but specifically a lot of what we do is cell-free synthetic biology. And so what cell-free synthetic biology is where we take the enzymes of transcription and translation out of the cell and use them to run gene circuits for gene circuit-based sensors, or as a platform for the production of proteins that could be therapeutic or lab reagents. Those are the two main efforts that we have in the lab, but we have other projects as well, but those are the big ones.
Okay, and over the past couple of years, the Pardee Lab has had a lot of great research occurring. Can you please describe some of the main goals of the lab?
Sure. So, as I mentioned, we build sensors, and so we do this using gene circuits that are sensitive to the specific sequence of your target. So, for instance, we have a project that is focused on the Zika virus, identifying the Zika virus in patient samples, as well as other arbo or mosquito-borne illnesses. And so we use the pathogen's genome as a barcode, and our sensor, which in this case is a toll-hold switch, is able to identify 30 base pair regions in the genome, and basically that, if it's present, we get a color change in these are paper-based reactions, so those enzymes of transcription and translation are embedded in paper, and in the presence of the right target, we get a change in color, which indicates sort of to the user's eye that there's the presence of the pathogen. And so we've applied that concept to, like I said, the Zika virus, and we have field trials and patient trials ongoing in Latin America. So we have our collaborators are in 5 different countries, 8 different labs, and our collaborators in Latin America are in Brazil, Ecuador, and Colombia. And there's actually a group down there right now in Brazil for a month from Aaron Wheeler's group, who's also at the University of Toronto, and his group is down in Recife, Brazil, testing their technology to extract the genomes, viral genome, out of patient samples for use in this technology. We've also applied the same sensor technology to many other pathogens and many sort of ongoing projects that hopefully you'll get to read about soon.
Thank you. And your lab has developed 2 biotechnologies. Can you explain what these biotechnologies are?
Sure. Right. So the one is the sensor. So this is where we've embedded the enzymes of transcription and translation into paper. We freeze dry it so that you can store and distribute this at room temperature. So you don't need a cold chain. And that is used basically as the sensor. And the other form is we freeze dry the cell-free system into pellets. And again, these pellets can be distributed at room temperature, stored. And then at the point of need or use, water is added back. And all the enzymes that were dried kick into action and start transcribing and translating the therapeutic of interest. And so in the past, we've published work where this system's been used to produce 4 different vaccine antigens. This work was done in collaboration with Jim Collins, who is my postdoc supervisor. And one of those vaccine antigens was tested in mice. It was the diphtheria DT5 antigen, and we demonstrated that with our system we could immunize basically or induce immunization in mice. But we've also used that production platform for reconstituting a 5-gene metabolic pathway to make small molecules, as well as antimicrobial peptides, sort of as an alternate to antibiotics, and antibodies, which are widely used in therapeutics, so as a targeting agent for therapeutics, or in labs as reagents in the lab. And so in that case, we could conjugate our antibodies to 6 different functional groups and turn them into tools.
And have there been any big hurdles in your work?
There has been, there has, yes. I would say in general, actually, that's our job, is overcoming hurdles, which can be difficult. I'm sure other people who are in the IGM program also recognize this challenge, but that's basically our job, right? It's the first time someone tries to do something, there's a lot of unexpected and expected challenges. And so what we try to do is tackle those challenges as rationally as we can, break them down into small parts to solve them. Some of the challenges that we're going through right now are in the field. When you're outside of a conventional lab or away from your home lab, how do you do science in those environments that are maybe more challenging? And so one of our solutions to that challenge was, I guess I'll start with one of the challenges. So one of the challenges is our collaborators in Latin America had 96 well plate readers, and we needed 384 well plate readers. So that may seem like a very small difference, but for scaling up reading patient samples and also the volume of reactions, it was really important that we had this smaller, more compact sort of form to work in. So these 384 multi-well plates. And so what we've done, or what groups of students in my group did, was they literally built their own plate readers, and now we have these plate readers in Latin America that are reading data and streaming it to the online database for the project. And so we sort of turned a challenge into an opportunity, and so we're hoping to share that work soon with publication.
And according to your lab's website, both of your biotechnologies are based on systems that use freeze-dried, cell-free biochemical reactions that are activated by simply adding water. Can you elaborate on what this really means?
Sure. So I'll break down the phrase. So freeze-dried, cell-free biochemical reactions. So if we start with freeze-dried, what we're doing is we're taking basically a biological soup, and we flash freeze it in liquid nitrogen so that it freezes very quickly, and that helps lock the proteins into one conformation, making them more stable. And then the process of freeze drying is where you remove water from that frozen sample without it going through a liquid phase. So it goes straight from solid ice to dry powder, and that helps keep things stable. And it's how a lot of food is distributed. So like ramen noodles or Lipton soup, packs can be freeze-dried, and medicine can be freeze-dried. And so we're just basically taking that concept and applying it to our biochemical system. The next word in that sort of description is cell-free. So that is where we're taking the contents of bacterial cells or mammalian cells or tobacco cells out of the cell itself. And that process is really important because it makes things biosafe. So you can take things now that would conventionally be a biohazard, right? So E. coli or some other organism that can self-replicate. Generally, those are very regulated, and so you can't take them out of the lab. But once we've created this cell-free environment, now we can run these gene circuits or expression platforms pretty much anywhere in your pocket. Put it on a plane, travel around the world, and do whatever you need to do with it. And so that's a big thing. So far we've got room temperature storage thanks to freeze drying. It's biosafe thanks to being cell-free. And then the biochemical reaction portion, this is where basically this is the machinery that does the work. So in these reactions, there's RNA polymerase, there's all the enzymes responsible for translation, there's ribosomes, and they all work in concert to transcribe DNA to RNA and then translate RNA to protein. And these cell-free biochemical reactions can either be recombinant, which means that each of these components has been purified individually and then transferred into this reaction. So they're very defined. Everyone knows exactly what is in those reactions. Or they can be just lysates, and those are much less expensive, and those can be made in almost any lab. And so those are where you basically just lyse cells and do some simple sort of wash steps and centrifugation steps, and you get your reaction. So both perform about the same thing. One is very defined and the other is much more crude.
And do you foresee, or is there anything that you would like to see in further research for these biotechnologies?
Yes. We've got quite a good group now working and on these problems, and there's a lot to do. The field of synthetic biology in general is growing rapidly, and the field of cell-free synthetic biology has sort of exploded over the last 5 or so years. And so there are a lot of problems to solve. Some are very technical, so those would be problems about sort of the nuts and bolts of what's going on inside, making reactions more efficient, more robust. And then there's a lot of other applications, which is sort of the area where we focus, where it's applying those tools to solve specific problems. So there's a lot going on in the field. There's a lot. We're doing lots. We'll be publishing on some of those soon. Others sort of, I guess, like if I pull back and look at the field in general, this gets a bit technical, but like things like, can you express whole IgG antibodies? So those are the ones that are naturally in our bodies, technically challenging to produce. And groups like Michael Jewett's group and others have started to tackle this in a real way. Other things would be post-translational modifications of the proteins. So this would be glycosylation of certain proteins, which is really important for their biological function. And normally E. coli does not perform this. So groups, also including Michael Jewett and others, are working to embed that capacity into bacterial cell-free systems. I could go on, but sort of the last one I'll talk about is the cell-free production of commodities and high-value products. So this has been the domain of cell-based synthetic biology and industrial biotechnology for quite a while, and I think it's interesting to think about moving that into the cell-free realm as well.
Okay, and the next question might be a bit repetitive, just of what we've already talked about, but I'll say it just in case there's like an expansion that could be made, or it can be like changed around editing or whatever. Sure. But the Pardee Lab is part of the Pan-American Zika diagnostic team, which is in relation to this second biggest, I'm assuming is the word, biotechnology currently in the works at the Pardee Lab. Can you explain what the Zika virus is and what the future goals of this diagnostic team are?
Sure. Okay. So the Pan-American Zika Diagnostic Team. This is funded by CIHR and the IDRC, which are both Canadian funding agencies, and it funds the Canadian side, and it also funds the Latin American teams. So it's a very interesting research or funding program, because normally that's not something that's possible, and has really made this project sort of possible. It also includes 2 American teams, Jim Collins at MIT and Beast Institute, and also Alex Green at Arizona State University, and they contribute to this project and the technologies as well. And so this project has 2 main efforts. One is taking the work that we've published for the Zika virus diagnostics and taking it out of the laboratory environment and making it practically sort of possible to perform diagnostics in the field. And so if you think about taking a patient sample like blood or mucus or urine, there's a lot of things you need to separate before you can get at the virus. And so how do we do that in the field in a way that's easy, simple, low cost? So how do we isolate the virus, amplify its genome, and then expose it to the test in a really low cost, robust way in the field? Another aspect of this project is, how does the technology work when it's exposed to real patient samples? How does it compare to the gold standards like PCR that are sort of the standard way to do these diagnostics right now? And then the final piece is to expand the reach beyond the Zika virus to other mosquito-borne illnesses that are at this point actually a much larger problem than Zika itself. The Zika infections have really tailed off, but there's a lot of other mosquito-borne illnesses that continue to be issues, and so we're addressing those. And so does that pretty much? Yeah, okay.
So what has been the most promising result so far that's showing like the potential of the system?
So, let's see, without giving away So, the nice thing is that the system works for patient samples. That's I would say, is the big home run, and we're sort of working on, like I said, Aaron Wheeler's team on how to sort of port this technology into sort of remote, outside of laboratory settings, and we're expanding now into other sort of pathogens as well.
And from a market perspective, is the economic efficiency of producing this portable and low-cost system to high pandemic areas already reached, or is there still room for improvement?
There's definitely room for improvement. So we're still at like that sort of, cottage industry level where we're just like, we're doing it, we're just a handful of labs using what we can get. And so the cost is still, while it's still low, it's still probably under $1 per test, it's still much higher than it has to be. And so I think as this technology matures and scale occurs, price is going to fall substantially, thanks to the work, obviously,